And so I think sometimes things you read in BMJ or from British authors, they're just very clear. And I was very excited a couple years ago when this article came out, because I think what they said about ITT was just so clearly written, so nicely written. And so I put it up here to read to you guys. It says, ITT gives a pragmatic estimate of the benefit of treatment policy rather than a benefit in patients who've received the treatment as planned. A full application of ITT is possible only when complete outcome data are available and all randomized. Many trials that claim ITT vary in their handling of missing data. And ITT is often inadequately described or applied. So I think these are kind of important messages about ITT that you should think about as you're reading trials. So, so many times people will read a trial and they say, oh, but ITT, it's perfect. Well, and the answer is it may or may not be. And so you really need to look in more detail. So they recommend ITT be thought of not only in evaluating a trial, but in designing a trial. That it's considered a complete trial strategy and not simply an analysis issue. And so in the design of the trial, they suggest that you justify in advance any inclusion which, if violated, will merit exclusion from the ITT. So if you're going to exclude people for some reason, at the very least, say that in the beginning. In the conduct of the trial, you want to minimize missing response on the primary outcome and follow up subjects who withdrew from the treatment. So those are all things we've talked about. In the analysis, you want to include all subjects according to the original randomization and investigate the potential effects of missing response. And you could do that by doing various types of, say, per protocol analyses or various types of inferences about the people that are missing. Assume the worst case, all the missing people died. The best case, all the missing people were alive and healthy. So these are some recommendations about how to deal with this. Also, in reporting of the results, they suggest reporting how the ITT was used, to report deviations and missingness, discuss the potential effects of missing response, and base your primary conclusions on the ITT analysis, even if the per protocol analysis maybe looks a little better. So to summarize all this stuff, the best trial we could do is a trial in which all participants remain on medications and all participants are followed for the primary outcome until the end of the study. So when you're designing your study, when you're evaluating your studies, this is what you kind of think is sort of the ideal. And also a pre-planned analysis and handling of deviations from the protocol would be an important factor. The design and the implementation of the study can help move toward the goal, so it's not just an analysis issue, it's something you think about from the beginning, and it's very important that you report the details of adherence and how it could affect results. So once again, that kind of summarizes all these things. So let me go on, and I've got about 15 minutes. I just have a few slides here. This is kind of going back to the last lecture. Some of the stuff is related to what we've been talking about. And this is what I talked about in the last lecture. And these are the things I didn't get to talk about. And there's one interesting example, which is why I want to kind of talk about this. So last week we talked about different statistical things. And one of those was adjustment for baseline covariables, which I mentioned but didn't have a chance to talk about. So this is something that what you say, I do a clinical trial. What if an important prognostic variable, for example, a confounder, is maldistributed by chance alone? Say, for example, we did a trial with a myocardial infarction as an endpoint. You get all done. You had bad luck with your coin flip, and the placebos are older than the people who are treated. So you're worried. We say, well, what if the placebos have a higher rate because they're older? So you might say, well, rather than just doing an analysis of intention to treat, which we just talked about, I'll do an analysis where I adjust for age, because the age is different. And you might, can you do that? So this is kind of strangely enough, a somewhat controversial issue. Because people have shown that if you have a null trial, if you just go in and start adjusting for enough variables, you can eventually change the results. So there's a high potential for what people in the technical term call hanky-panky. in a trial. So you can really manipulate the results of your trial just by going in and trying to adjust for this, adjust for that, adjust for these two things. So it's not quite as benign as it sounds. So I think it's not to say that it's not valid, but it's something you need to do carefully. And so the solutions that I think are generally accepted, that if you want to adjust, that if a specific variable is highly prognostic, for example, age and MI, then use that to stratify in your randomization. So we talked about stratified randomization in the first lecture. So if you believe age is really important, you might stratify your randomization into those above 70, for example, and those below 70. If you're going to perform an analysis, do it unadjusted and then adjusted and see what difference it makes. And in reality, if you have something that is maldistributed based on chance alone, it's seldom that it really makes a difference. But there are some cases when it does. And then most importantly, I think, and again, the theme that I always bring up about almost everything is to pre-specify what you're going to do. So pre-specify a condition under which an adjustment will be done for a small set of variables. For example, before you do the trial, say you decide not to stratify, but you say in advance that this is a trial of MI, and I know that age, blood pressure, and LDL are very prognostic. So I decide in advance that if any of those three variables are maldistributed at baseline, defined as, say, a p-value less than 0.05 between treatments at baseline, then I adjust for those variables only. So that's a very typical example of a kind of pre-specification that gets rid of this sort of multiple comparisons issue of doing all possible baseline comparisons, all possible covariate adjustments and seeing which one works. Okay, so that was adjustment. Now, what about multiple efficacy endpoints? So you may have multiple endpoints in your clinical trial. We talked about this last time in the SELECT study, the example. They had prostate cancer, all cancers, prostate cancer mortality, and that induces multiple comparisons problems. So if you have a lot of different outcomes, you got to be careful how you deal with them and how you deal with them statistically. Some solutions are very, very explicit predefinition of endpoints. You certainly don't want to say, I'm going to look at all kinds of cancers and on all kinds of individual cancers. You don't want to do that. So you might want to say, I'm really interested in prostate cancer and all cancer, or these three, to kind of limit the number of endpoints. You could also do a formal p-value adjustment. Say if you have two endpoints, say you had all cancer, prostate cancer, you could do a Bonferroni and do each of those at a p-value of 0.025 because you're doing two tests. Now, if you're doing a study for the FDA for drug approval, they require a single primary endpoint. So you have to explicitly define what that endpoint is, be very clear, and that is what it's going to determine if your drug is approved or not. So if you don't show an effect on prostate cancer, but you do on all cancer, and you've specified prostate cancer as your primary outcome, the FDA will not approve your drug. And there are lots of examples where it's probably not sensible, but that's the way they operate in order to keep control of this type 1 error, potential type 1 error problem. If you go into the FDA and you have two primary endpoints, they'll let you do that, but then you have to somehow adjust your p-value. So either do a Bonferroni, very commonly done, or you can do some kind of like nested hypothesis testing where you look at one of the primary outcomes and you only test the second if the first is significant. So this is a real issue when you have multiple efficacy. And a great example that I always have is the Moore trial, where it was a trial of raloxifene, and the main outcome was vertebral fractures, and the secondary outcome was non-vertebral fractures. And then they had 13 different types of non-vertebral fractures. And overall, the effect on vertebral fractures, the main outcome was fine. But when they looked at non-vertebral fractures, there was no effect overall. And it turns out there were 14 subtypes of these non-vertebral fractures, and there was one, ankle fractures, that were significantly different. Well, that's at p of 0.05, and they have almost 20 tests, so you'd expect by chance alone you'd have one significant finding. So they wanted to title the paper, Raloxifene Reduces Ankle Fractures, and luckily the investigators who were involved were strident enough that that didn't happen. So that would be kind of a worst case. Then in terms of the analysis, this is again from WHI. I think I may have showed this last time, invasive breast cancer. We might wonder what's NCI and ACI.